If you could clone your most beloved pet, would you do it? If you could help the environment by mixing the genes of different species, would that be right? And if you could bring back an animal that's been dead for thousands of years, should you try to? So far in this series, we've explored how biotechnology is transforming food and medicine.
Thank you very much.
We've met a mouse with a human ear on its back, rabbits that glow in the dark,
Oh, yes.
and featherless chickens. But this week we go one stage further, to the outer limits of what science can achieve. This is not tomorrow's world. It's happening now. If all the creatures created by science were gathered together on one farm, then this is what it would look like. The farm is imaginary, but everything you will see on it is real. Welcome to our animal farm. We've chosen two people to explore the farm. Olivia Judson wants to persuade you that biotechnology is to be embraced.
I'm a biologist, and I've come to realize that biotechnology is affecting all of our lives, whether we are aware of it or not. It's already changing our food and our medicine, but it could also be used to improve the environment or correct past mistakes, or even satisfy more frivolous desires. I find this technology absolutely amazing, and I'm fascinated by the possibilities.
But where Olivia's all fired up, food writer Giles Corran finds it disturbing.
I like things on the farm to be done as naturally as possible. I'm worried that once we start genetically modifying things, once we start messing with nature, we run the risk of creating terrible problems for humanity in the long run. After all, can the scientists be trusted to do the right thing with all this newfound knowledge of theirs? And most frighteningly of all, once the secrets of my genes have been unlocked, where does that leave me?
On our final visit to the farm, Olivia and Giles are journeying to the furthest frontiers of biotechnology. Every living thing on the planet has been created by a series of instructions known as genes. One particular set of these instructions makes trees, another makes cows, still another makes humans. But now scientists have discovered it's possible to shuffle the pack. We can take a gene from one species and put it into another, creating extraordinary new creatures that work for us in ways we've never seen before. And Olivia's about to meet one. It's part mammal, part arachnid.
I've always liked spiders. They are so beautiful, elegant, and strange. And their silk is one of the strongest substances known to man.
Oh, you can see a bit of silk hanging out behind.
Dragline silk, which a spider uses to make the spokes of its web, is tougher than Kevlar, stretches better than nylon, and weight for weight is 5 times stronger than steel.
She's got such lovely hairy legs.
It's believed a cord of silk just a few centimetres across could lift a jumbo jet off the ground. But despite these remarkable properties, it's always been impossible to produce spider silk on a commercial scale. Unlike silkworms, spiders are too antisocial to farm successfully.
People have actually tried in both Britain and France in the 1800s to grow spiders in barns. And they're very territorial, and they actually will kill each other to get enough territory.
But now Professor Randy Lewis thinks he's come up with a way to change all that.
What we've done is taken the gene for spider silk from a spider, and we've incorporated that into the genes of a goat. And the goat produces that silk protein in its milk.
Yes, you did hear that correctly. He has put a spider gene into a goat.
So how do you take a gene from this creature and put it into a goat?
Well, in this case, because we want to make sure that we had the right gene, since they make different kinds of silk, we actually went to the gland that produces the dragline silk. and we extracted the gene from there that was actually being made, so we knew it was the right one.
To create a spider goat, Professor Lewis took the gene that allows the spider to make silk and put it into bacteria. As the bacteria multiply, so does the gene. The bacteria are then injected into the egg of a goat. When the egg develops, the gene allows the new goat to produce the proteins that make silk.
You don't notice that there's anything peculiar about the goats in any other respect?
We don't see any differences in the goat, and the goats behave absolutely normally.
Extracting the silk from goats is a tricky process. The spider gene allows the goats to produce the proteins that are the building blocks of silk in their milk. There are many other proteins in milk, and this new gene adds just one more. Scientists filter the goat's milk to isolate the silk proteins, and then comes the delicate part.
How do you actually make it into fiber?
So what we've done is loaded the syringe with the spider silk protein in solution into this particular syringe. What will happen is that the instrument then very slowly pushes down on the syringe, which forces the silk solution into an alcohol. What happens is as the fiber starts to form, then we can actually pull it. From here and take it up out of the solution and then start winding it on these winders so that we have a silk fiber that we actually can begin to test.
It looks like a sort of a giant version of the spooling part of my mother's old sewing machine.
Very similar. We spool it on to collect enough silk that we can actually do testing.
For the first time ever, it might be possible to make spider silk in bulk.
From a liter of milk, we can actually get about 7 grams of silk, and at about 100 meters per milligram, we're talking about in excess of 70,000 meters of silk.
So I seem to be holding a piece of spider silk that has come out of goat's milk. It's extraordinary. It is actually quite stretchy. But it's fine, like the very finest hair on a baby's head.
The technology is pretty nifty, but why would we want to produce spider silk on an industrial scale?
I think things divide into a couple categories. In the medical field, people are particularly interested in artificial ligaments and artificial tendons. And people are interested in it for body armor. Particularly the military is very interested because we estimate that we could make bulletproof vests that would be about 1/3 the current weight. So somewhere around 5 pounds versus 16 pounds of the current ones.
It says so much about the fundamental evolution of life that you can take a gene from a spider and put it into a goat, and it works.
And it works perfectly.
It's amazing.
The spider gene only operates in the goat's milk glands. But the next step for scientists was to make a GM animal where the new gene was switched on in every cell of the body. They had to choose a gene that would show up wherever they put it. So how could they tell it was working? Simple. They could see it. Welcome back to our imaginary farm in which all the inhabitants have been created by science. Olivia Judson has already seen goats with a spider gene.
It's extraordinary.
But this gene was only working in one specific place. The next step for scientists is to make a creature where a new gene is switched on in every cell of its body. And Olivia is heading to the pigsty to see if it can be done.
The first thing you notice about these pigs is that they've got a distinct yellowish tinge. And if we turn out the light, they glow bright green. These animals have been created by Professor Wu Xin Chi. He's inserted a gene from a jellyfish into the embryo of a pig. And the results are remarkable. These animals take genetic modification to a new level. They're the first pigs to have a new gene working in every single cell of their bodies, meaning they have not only green skin, but green lungs, livers, and hearts as well. So if you're a medical researcher studying, say, bone grafts, you could now take bone cells from a green pig and put them into a normal pig, and you'd get an incredible new level of detail about how the transplant process works. So although they may look bizarre, these pigs haven't been created to be sideshow freaks. In fact, they're rooting around at the foremost frontiers of medical research.
Once they created an animal that glowed in the dark, scientists realized the fluorescing gene had wider uses than just medical research. They decided to see if they could make the gene switch on and off in response to the animal's environment. And Giles is about to find out if they did it.
Apparently, these fish are normally a pale, fleshy colour. But just like the freaky glowing pigs, they've had a gene put into them which makes them fluoresce. Only this time it's not from jellyfish, it's from coral.
The gene is taken out of the sea coral. Can be done with the jellyfish gene, but these are done with sea coral. And as soon as the egg is fertilized, the gene is added to the egg, and then it integrates into the DNA.
These fish were originally developed as a kind of early warning system for toxins in water. The idea was that you drop them into a bucket of water taken from a river, and if the water was polluted, then the gene would switch on and the fish would start to glow. So is that what they're used for now?
The scientists are still working on the switch to detect pollutants. At this point, we're currently using them in the ornamental fish market, otherwise known as the pet fish market.
So they're just pets?
Absolutely, and use them in their house for enjoyment, stress release.
As it turns out, these stress-relieving toys are the first genetically modified animals on sale to the public anywhere in the world. Does this mean we could be looking at a future of glow-in-the-dark pet shops?
Fluorescence marker gene technology works with all cells and organisms up to people. There's a green fluorescent rabbit. There's been a monkey that was green fluorescent.
Why don't you sell those in pet shops?
We have not gotten beyond fish. I mean, we're looking at a specific market, and that's really a business issue.
A business issue? We're talking about the building blocks of life. We shouldn't be messing about with this stuff. What if the fish somehow get out? So presumably if these fish escaped and they mated with other fish, you'd end up with lots of glowing fish. I mean, they're not going to mate immediately with a shark, but gradually with things that are a little bit bigger. Until, are we looking at a situation in a few hundred years where every fish in the sea glows in the dark?
You have to look at this in evolutionary terms. Evolution hones organisms over millions of years. This fluorescence gene has no purpose in an evolutionary fitness perspective. It causes a significant burden, simply predation. I mean, a predator, this is a bright red neon or green or orange sign that says, "Eat me." These fish are ecologically weak, so they would be cut out of the gene pool. So if it were to get out, it would die.
Oh, really? Well, I'm not entirely convinced. When it comes down to it, are there actually any guarantees?
Anything in the world that we do has risk, and I can't tell you for sure that these fish are never going to get out. Any type of biotech animal has to be considered on a case-by-case basis, just like we consider pharmaceutical drugs, for example. We have certain processes that we go through, and we feel a certain comfort level that they're safe.
So Alan can't promise me that these new genes won't get out and cause havoc. And this is the nub of all my concerns. Do we really understand what we're doing here? In fact, genes from some GM organisms have escaped into the wild, so I reserve the right to be a bit worried.
While Giles is concerned about GM and its impact on the environment, what some call genetic pollution, Olivia has a different take.
I find it depressing that people assume if you care about the environment, you can't support genetic modification. In fact, a lot of scientists working in the field are so concerned about the environment, they're creating animals specifically to help it. I like bacon as much as the next person, but the amount of pork we eat is causing serious problems. Last year, we killed more than 2 billion pigs, and these creatures provide more dietary protein, more cheaply, to more people than any other animal. But all this consumption comes at a cost.
Animal agriculture is a major consumer of the natural environment. That is, the impacts on the atmosphere, upon water utilization, and in particular, pig agriculture or hog agriculture produces copious amounts of manure.
Manure, it's a natural product, but on an industrial scale, it's a serious environmental pollutant. And poo from pigs is particularly toxic.
Pig manure has a very high level of phosphorus within it from normal diets through normal pigs. And that phosphorus usually is more than is required by crops produced on land fertilized with pig manure.
Pig manure is packed with phosphorus because pigs simply can't digest it. And this excess is causing some serious damage. It runs off into the water supply, causing the overgrowth of algae and killing fish. It seems an impossible problem of biology, but John Phillips has found a way to fix it.
A very simple and effective solution to the environmental problems caused by high phosphorus pig manure found right behind these doors.
To tackle the problem, John has created something he calls the EnviroPig. This animal looks normal, but it's anything but.
These pigs are unlike any other pigs in the world. They're different because they carry a very special gene that allows them to digest all of the phosphorus in their diet. The gene is constructed in the laboratory that we use.
The remarkable thing about the EnviroPigs is that they carry a man-made gene, which John has created from scratch.
One part comes from E. coli bacteria, which produces an enzyme that can break down phosphorus. And the other comes from a mouse, which produces a genetic switch that allows this special enzyme to be made in saliva. These two bits of DNA were combined to form a new compound gene, which was then inserted into a fertilized pig egg. Now every time John's pigs eat and produce saliva, they also make the phosphorus-busting enzyme, helping to make their poo more environmentally friendly.
The aim, ultimately, we think, is that the global environment would be best served if every pig could digest all of its organic phosphorus in its diet the way these pigs do.
So John wants every pig in the world to be an EnviroPig. I think it's an idea with merit, but he's encountered a lot of obstacles.
So there are many people out in the community that do have concerns about transgenic changes in animals, and some of these people have gone about intentionally frightening the public about this technology. So we have an uphill battle in terms of people's perceptions. This is an effective way of dealing with a serious environmental problem. It's simple, it can be propagated very rapidly. Any other technology cannot touch this in terms of the rapidity of its application on the farm or the extent of the change that can occur as a result of the technology. Nothing comes close to this.
I think it's worth saying again that pork consumption plays a vital role in maintaining human health across the globe. But as long as we keep eating pigs at this rate, we're causing problems for the environment. So we face a decision. Can we accept such a radical solution? I think we should.
Once scientists have gone to the trouble of improving on evolution, either through GM or traditional breeding, the next question is how to preserve their success. Here on the farm, we've got someone who's found a way to hold on to perfection. For his next stop, Giles is off to the stables to meet a world-famous sporting duo who've just made scientific history.
He's $1 million. He's a Kentucky Derby winner.
Here on our farm, we've gathered together some of the most extreme creatures that science has produced. So far, we've seen how genetic engineering can be used to produce animals to our exact specifications. But once you've made the ultimate creature, how do you ensure you can preserve it? Giles is heading to the stables to find out.
Hi.
You're Charmaine then.
Hi.
And
This is Scamper.
So what's so special about this horse?
He's a 10-time world champion barrel racing horse, and he won those 10 championships consecutively.
And what is barrel racing exactly?
Barrel racing is a timed event around 3 barrels.
Charmaine Rodman, Gulf, California, on the Scamper. Come on, Scamp. Let's go, bay horse.
Who was you riding him?
I was the only one who rode him.
He still can at 14. He's $1 million winner.
Scamper was a bronc when he was young. And
bronc?
He bucked.
Wild,
he bucked a lot of people off. There's no telling what kind of abuse he had, being that he was so mean. He went through 3 or 4 sale yards. Nobody wanted him.
So Scamper had a troubled childhood. But just when everyone else had given up on him, Charmaine came to his rescue. It's like Seabiscuit. It's a proper romance.
Yeah, and I was a little 11-year-old girl that just was horse crazy, and he met me for the first time, and we just basically fell in love. It was a match made in heaven, love at first sight. Scamper made me who I was and what I was, and he did. You know, I lived every day of my life with him for 10 years while I was winning a world championship, never left him.
But all good things come to an end. So when did Scamper stop racing?
He's been retired for around 10 years now.
So when he was retired, did you carry on racing on different horses?
Yes.
And they weren't as good?
Oh, no.
What was it they didn't have, the other horses?
They didn't have whatever Scamper was made of.
Scamper's a world-famous horse, and in theory, his genetics, basically his sperm, are worth a fortune. But in fact, it's more complicated than that. And Scamper never had any children?
No, he's a gelding.
Oh, poor fellow. And when you met him, he was already a gelding?
He was already a gelding.
So what do you do when you've got $1 million racehorse and no way to reproduce him? Well, here on the farm, there's an obvious solution.
So you can't have children, so you've decided to clone him?
We decided to clone him so we could get that same genetics.
How did it come about? How did they clone Scamper? They took cells from him, or?
Yeah, they took a biopsy, just a little small amount of tissue from Scamper, and of course we gene banked him, and then when they had the science to...
You gene banked him before they had the science?
Yes.
And then what, they gave you a call when they had the technology?
Yes, when they knew they had it right, they said, okay, we're ready to go.
The tissue that Charmaine gene banked contained DNA with all the instructions necessary to make Scamper. This DNA was then extracted from one of the cells and injected into an empty horse egg. The egg was given an electric shock to trick it into dividing and implanted into the womb of a surrogate mare. The resulting baby was a precise genetic copy of Scamper, exactly in the way an identical twin would be. The little clone is named Clayton. And Scamper's about to meet his younger self.
There's Clayton.
Oh. Well, he looks pretty normal, but this is crazy stuff, and I reckon it must be even weirder for Scamper. Have they met many times before?
This is the second time they've met. And Scamper's not real fond of Clayton. He likes his mama.
Really?
Yes, so he's protecting the mama from the baby.
Is he? Is that that's a man thing?
That's a scamper thing.
Is this chopping mouth, is that a threat?
Yes.
So Scamper doesn't think much of his clone. But what about Charmaine? You see similarities between the two already.
He carries himself the same. He has the same arch in his neck. He's just very confident. The first time I saw him, I just couldn't take my eyes off of him. It just makes the hair on the back of your neck stand up to think that this is how Scamper was when he was a baby, and it's real emotional.
He doesn't look identical. I mean, maybe is that because he's young? Is that what Scamper looked like when he was a kid?
I didn't see Scamper when he was a baby, but conformation-wise, the shoulder, the hips, everything looks the same. From what I understand, the white migrates in the womb, so that has nothing to do with the DNA.
Clayton's been created to do what Scamper can't, produce sperm, which will then be sold to other breeders. But what if it was Scamper's tough childhood, more than his DNA, that took him to the top?
We obviously don't know how much of his past life made him try as hard as what he did. Kind of like the kids that grow up in a bad part of town and they knew they had to overcome a lot. Kind of the same thing.
I guess only time will tell. But while Charmaine is delighted about Clayton's arrival, not everyone is thrilled that he's here.
We've had a few negative comments.
Such as?
I think that we're playing God. And now that we've created Clayton, now what if they clone Osama bin Laden?
That's what they said?
Yes.
God, that would be scary. But he wouldn't be much good at barrel racing, would he, Osama? In spite of the skeptics, Charmaine's certain she's done the right thing.
I think the people who really don't understand the science behind it may oppose it, but the reasons I did it were, yeah, I love Scamper, but I have a passion for great horses and their ability. And I've struggled with other horses past him that had so many physical problems from competing that it's hard to get past all that. And if we can start breeding some horses that stayed as sound as him, then I think we're doing a real noble thing.
I think noble might be pushing it, but the possibilities of this technology are extraordinary. Where will it end? Are they going to give Shergar another shot at the Derby? Could Red Rum really ride again?
In fact, that's not as far-fetched as it may sound. Because once they discovered they could clone live animals, scientists realized they didn't have to stop there. For Olivia's next appointment, let's send her to the outhouses to meet a man who's giving cloning a whole new twist with the farm's very own 21st century Noah's Ark.
Seltzer.
Oh, wow. So what am I looking at?
Well, we have the cells all in their little high-rises here, so we can find them.
I believe we're in the middle of a mass extinction event. Over 16,000 plants and animals are on the threatened species list. Some scientists estimate that because of human activity, 50% of all life forms may become extinct within the next 100 years. Faced with this crisis, Oliver Ryder has come up with a desperate solution. He's collecting the DNA of as many endangered species as possible and freezing it.
Just received whale tissues. There's...
Oh, yes, blue whale.
We also have multiple vials in here of, I believe this is an Indian rhino.
Oh, wow.
So all of these cells, and I'm going to put them back in so they don't warm up, are stored in this inventory system, and we can hold tens of thousands of samples in a freezer like this.
In a pretty small space.
Yes, it's very efficient.
It looks like a sort of witch's cauldron. Oh, it's quite dramatic.
It's time to tuck them back in.
So in one of these tanks, we have tissues from, say, gorillas, rhinos, orangutans, whatever.
hippos, giraffes, dolphins.
Is it all mammals, or do we have ants and bees and wasps and...
We don't have any insects. Recently, we've been adding reptiles and birds. For example, California condors. We'd like to add amphibians. There's nobody saving amphibian genetic resources like this. So we have about 600 species represented in the tanks. But it's a very small part of biodiversity when you look at the number of species that are recognized to be endangered.
So how does an animal get into Oliver's Ark?
From mammals, we get a small snip of skin, and we can take that piece of skin, say that's half the size of a split pea, and take it into the lab, make a fine suspension of cells, and then we can freeze those cells after special cryoprotectant procedures, and they're in suspended animation. So 20 minutes later, after we take out a sample, it'll be growing again.
That sounds great in theory, but does it actually work? Well, it turns out Oliver's already tried it, using exactly the same technology employed to make Clayton: cloning. The survival of any species depends on it having a large breeding population, which creates a healthily diverse gene pool. But if this pool gets too small, the species can become vulnerable. One animal that's currently battling this problem is a rare breed of cow called the banteng.
The banteng populations in captivity are small. It's not possible to move animals from Europe because of mad cow disease, and so we have even an unanticipated event that's further shrinking the ability to manage a global gene pool, and the banteng become more highly inbred and less genetically diverse and more susceptible to extinction.
By a stroke of luck, Oliver discovered that banteng cells had been preserved in the frozen Ark. He decided to use them to inject vital new genetic diversity into the species by cloning an animal of supreme genetic importance.
This individual's valuable genetic contribution was thought to have been lost because the individual died 25 years ago.
In effect, Oliver has staged a resurrection. And more extraordinary still is the way he did it. Cloning requires an empty egg, but rather than use a banteng egg, Oliver decided to look to another species. He found that he could use an ordinary cow egg to house the banteng DNA, and a female cow to carry the banteng embryo. 5 months after she was implanted, the surrogate cow gave birth to a baby banteng.
So I'm quite intrigued by the fact that you can take a cow egg and put in the genetic material from another species and it works. Were you surprised by that?
I'm totally amazed. I, in fact, am on the record as saying it wouldn't work.
The banteng was created 4 years ago. The process was so groundbreaking, I was worried he might not have survived. I was glad to find out I was wrong.
So who are we looking at?
Right up close and personal is Jahava.
He's got a fabulous tongue.
He's quite special to us, not only because he's Jahava, but also because he's a cloned banteng.
And will he be allowed to fornicate with his companions?
Oh yeah, and we see him trying from time to time.
Oh, he's practicing.
He's practicing, that's it.
Excellent.
Now that we've cloned the dead banteng, similar technology could, perhaps, be used to bring back animals which haven't been seen on Earth for thousands of years. A saber-toothed tiger, or a woolly mammoth. But this is where Oliver draws the line.
In order to really bring a species back, you would want to bring back its gene pool, and there are not many species for which there's enough bank material that one could do that.
It'd be pretty cool if you'd have a woolly mammoth. That would be pretty cool.
No.
No?
See, I would be in the forefront against it.
Really?
Yes, it's a stunt. If bringing it back is something for our own amusement, I think that's the wrong reason.
In his fridges, Oliver keeps his Noah's Ark of the world's endangered animals, hoping it could save them from extinction. It's a valiant attempt to save part of the vast genetic heritage that we're destroying.
I think if somebody in the future would ring me up and I can say, what do you think we should be doing? I think the single most important thing that I come up with is they would say, please save more stuff for us. In the freezers, in the wild, whichever you can save, save it. And I'm convinced they'll be glad we did.
Inside each and every one of these test tubes is a creature waiting to be brought to life. Thanks to the frozen ark, could extinction itself be a thing of the past? Genetic research is affecting species from all over the animal kingdom, and here on the farm, we've seen many of them. But there's one creature that interests the scientists above all others. For his final meeting, Giles is heading to the basement, where experts are unlocking the genetic mysteries of the most powerful animal on Earth.
Genes play such an important part in our life, how we grow up, the kind of persons we become, and that is really that's the challenge for genetics in the future. It's also the excitement that we can better understand ourselves as individuals.
Giles is about to enter a place where they're putting animals to work in a mission to understand humans. The creatures being studied are so valuable, he must don protective clothing to ensure he doesn't contaminate them. In this labyrinth, mutant mice are being scrutinized for their genetic secrets. And there are literally thousands of them.
The mouse is a good model for the human, although it's small and furry, and we're big and we're not very furry. We know that its physiology, its biochemistry, the way it works is very similar to humans.
It's like Mouse City. Look at them all in their high-rise blocks. Apparently, mice and humans share over 90% of their genes, and for these geneticists, that's remarkably useful.
We know that in the human population, we're carrying all sorts of alterations in our DNA. You're carrying alterations in your DNA. Those alterations will make you more susceptible in the future to Alzheimer's disease, cardiovascular disease, diabetes as you get older. We need to know the relationship between those alterations and what's happening in the human population. So we can study how genes cause disease in mice, and we can then translate that information into working out how genes cause disease in humans.
To find out how genes work in mice, the animals are first injected with a chemical that randomly alters the DNA in their sperm.
All these male mice here have received an injection, a course of 3 injections in fact, that damages their sperm. So when their sperm actually regenerates, all their offspring should be carrying mutations.
The male mice with the mutated sperm are now placed in cages to mate with females. When the offspring are born, each is carefully inspected to find out what unusual new traits it might be carrying.
He does look quite normal. With the full screen, we will actually check that their teeth are there, they've got the right number of toes, the right number of tails.
Right.
And
do they often have more than one tail?
Not often.
The mutations in the sperm are totally random, so no one can predict what kind of mice they'll create.
She's actually got quite wonky ears there.
Once an unusual feature is discovered, the scientists look for the gene that's causing it. And it seems they're searching for everything, from genes that cause cancer to genes that cause dominant behaviour. But my favourites are the mice with a more specifically human flaw. Right, so these are the alcoholic mice.
Yes, these are the ones that are on the alcohol preference testing.
And they have a preference for it?
Yes, you can see, I don't know if you can see in the cage that there's 2 bottles. One of them is with ordinary water, and one of them with medical grade, well, you can see on there, 3% alcohol.
Which is quite high, 3%. Scientists believe that part of the reason people become alcoholic is genetic. These little fellows are on the equivalent of 2 bottles of whiskey a day.
This is actually a preference test, so the mouse itself has a choice.
Right.
Does it want to go to the water bottle, does it want to go to the alcohol?
As do alcoholics, frankly.
Yes, yeah.
To find out why the mice drink, They are anaesthetized and placed in a brain scanner. Researchers discovered the animals lack a crucial piece of neurochemistry, which leaves them feeling jittery and irritable all the time. And it's believed that drinking alcohol makes them feel more relaxed. The gene that causes this problem has been discovered, and the impact of the finding could be enormous.
There's no doubt that if we understand more about the relationship between gene and disease, we're not just understanding the symptoms, we really understand the mechanism of what's causing a person to drink too much alcohol.
And this kind of research could lead to a new era of personalized medicine.
In the future, a GP will take a blood sample from you. They will look at all the genes that might be involved in all these diseases, see whether you're carrying alterations in your genes that say you're at risk of disease. And then there's enormous opportunities to change your lifestyle, or perhaps even to intervene with drugs to ensure that we put off the day when you might develop those diseases.
They really have got everything down here. Just round the corner from the alcoholics are mice with an even more topical condition.
Nice to meet you.
Nice to meet you. What have you got here?
We've got some obese mice. So if I get these out, so these are brothers. They're about 14 weeks old. And so this one is about 50 grams, and his brother is about 30 grams.
Gosh, so what with me being 12.5 stone, that is like having a 20 stone brother?
Yes.
I would not hang out with him in public.
These mice have a genetic mutation which means they can't tell when they're full up.
He's actually burrowing into his food up to his... all he can see is his huge arse.
As a result, they just carry on eating.
This place is in Aladdin's cave of understanding, directly relevant to us all. It's incredible to think that because of genetics, future medicine could be tailored to our individual needs. But one thing's worrying me. Once we've identified certain characteristics by their DNA, won't societies that view those characteristics as undesirable be tempted to try and eliminate them?
I think most people regard actually treating disease by manipulating our DNA as a route that we don't want to go down, and a route that we don't need to go down yet. We know because of the complexity of the genome that messing with it is a dangerous and difficult business. What we want is to understand the relationship between genes and disease so that we can intervene, not at the level of the gene, but at how the gene is affecting the biology of that particular person, and how we can intervene with drugs or even just better understanding of whether they're predisposed to a particular disease so we can give lifestyle advice.
I hear what Professor Brown is saying, but scientists are currently trying to identify a gay gene, a musical gene, even an infidelity gene. One day they may find them, and when they do, what then?
For Giles and Olivia, the tour around Animal Farm is coming to an end. No one else has ever encountered so many different creatures created by science. From the featherless chickens to the cloned horse, the green pigs to the spider goats, they've been face to face with the extremes of genetic research. And now they've seen it all. What do they think?
I've seen so much. I had no idea how far things had gone. Some of the advances, medically speaking, are fairly miraculous. I met the girl whose bladder had been grown in a lab.
I can do whatever I want without having to worry about having accidents or having to go to the bathroom.
And I'll never forget those mice that could regenerate themselves.
And we actually did some interesting studies in spinal cord regeneration. So we cut the spinal cord completely, and in 3 weeks, they heal almost perfectly.
Going to see sheep that have been given human stem cells so that they have bits of human organs, I was really quite startled by that.
But what about the GM cow with the human immune system?
A cow with a human immune system, well, I mean, that's not actually very different from a cow with a cow immune system. It would be very helpful in fighting disease outbreaks.
OK, I agree there are great benefits for health.
The area where still I'm worried is food.
I thought that the Belgian Blues, although it was only selective breeding, I'm aware, and it wasn't an example of evil GM, nonetheless, prime example of why men shouldn't meddle. I just don't think it's necessary. I think it's just another tool for the wrong kind of farmers to wreak the wrong kind of havoc on the food chain.
Is it healthy? The animal is as healthy as another intensive producing animal.
And what about the super salmon?
I hated the super salmon, genetically modified to grow bigger, faster, quicker. That was another horror story.
How about golden rice?
Well, that's definitely a good thing. Genetically altering a staple crop so that it makes beta-carotene, which helps to save lives. It would be hard to argue against that.
So the question is, has the journey changed Giles's views?
The difference between me now and me before is that I now have some knowledge. So before, I just thought of cloning and transgenesis and all those things as being this scary thing which was just going to happen uncontrollably and that we couldn't do anything to stop it. Now, I don't think that. Now I understand to some extent what the scientists are trying to do. And having understood some of the science, it's enabled me to be less scared of some of the things that they do.
Well, it sounds like you've been converted.
I think the little knowledge that I have gained has given me a more even view of the project. I'm not quite so desperate to lynch the scientists.
Olivia was always positive about genetic modification. But did anything stand out for her?
I was surprised by how advanced a lot of the technologies are. I mean, I think that it's amazing that we have reached an understanding of nature that is so sophisticated that we can actually do these experiments. To me, the more I understand about biology and the more amazing I think it is. And you know, knowing more about how a robin sings helps me, you know, I enjoy the song of the robin more. And the idea that you can take a gene from a jellyfish and put it into a fish or a rabbit, and that the gene works, sort of gives me a sense of the immensity of evolutionary time, and so therefore a gene from a jellyfish would work in me. I think it's amazing.
One thing is certain. The genie has been let out of the bottle, and genetic science is here to stay. Our animal farm is only the beginning.
If you missed any of tonight's Animal Farm, you can now watch it with 4 On Demand's free catch-up service. Just go to channel4.com/4OD. On More 4 right now, Carol Thatcher's off to the Falklands to see her mother's legacy for herself in Mummy's War. Stay with Channel 4 though, Eddie's getting too close for comfort in ER next.